Hello, this is Ivan Rich. I'm CEO of HemoGenix, and welcome to this video on 50 years of colony-forming cell assays for hematopoietic cells. We have several videos on the HemoGenix YouTube channel on the colony-forming cell assays, but this year is a special year. 2016 marks half a century since the colony-forming assays were first published by Bradley and Metcalf working in Melbourne, Australia, and Pluznik and Sach working in Rehovot in Israel. It is safe to say that without the CFC assays, our knowledge of hematopoiesis would not be where it is today. It's also safe to say that our knowledge and understanding could have been even further advanced in several applications such as stem cell research, cellular therapy, and drug development if more attention had been paid to the information this assay can actually deliver. This slide gives you a short history of the early days of the CFC assay. If you're new to the field, or even if you've been working in the field for some time, you have probably been using a single product and you may not be aware that many other products are available to study hematopoietic stem and progenitor cells. In fact, as you will see, not all CFC products are created equal. Indeed, there are significant limitations to the CFC assays as we know it. So this video attempts to broaden the horizon and compares the different CFC assays available and how they can be used to further your research and advance the field. Now, I've been doing colony-forming assays since 1973. I actually learned it from Norman Iskov. So I have quite a bit of experience in this area. I also realize that some of the concepts and principles may be new to you. So I hope this will be educational as I compare different types of hematopoietic assays. The formation of colonies of cells is called a clonal assay. The primary use of a clonal assay is to identify the functional ability of primitive cells that are so few in number they cannot be morphologically identified. So a clonal assay is a functional assay. The main advantage of using a clonal assay is that the cells growing outward from the colony-forming cell differentiate and mature, thereby identifying the original cell that gave rise to the colony. However, only cells that have the ability to proliferate can be identified in a clonal assay. Although a clonal assay requires cell proliferation, a clonal assay does not directly measure cell proliferation, and this is a very important and fundamental concept. Proliferation and differentiation cannot be measured using the same readout or endpoint. Proliferation always occurs prior to differentiation, and although this might seem obvious, it is often forgotten or dismissed. Once again, a CFC assay does not and cannot directly measure cell proliferation. Certainly the cells need to proliferate to form a colony, but that is not what you're measuring in a CFC assay. And this has been propagated for decades and is a false assumption. In fact, all stem cell systems are based on the interplay and difference between the proliferation and differentiation processes. You may have seen diagrams like this of the hematopoietic organization and hierarchy. It emphasizes the fact that without the CFC assay, it would have been very difficult to understand the system, which I like to compare to a tree. The primitive stem cells form the roots, the mature stem cells the trunk, the progenitor cells the branches, the precursor cells the small branches or twigs of the tree, and the leaves the functional mature cells. With the identification of different growth factors and cytokines, it's been possible to detect different cell populations, and I'll describe this a little later. The range of the CFC assay is shown on the left, and because the assay can be used to identify individual colonies, its range can be extended almost to the mature cells. However, it is not that sensitive, so it's quite difficult to accurately study some of the principal functions of stem cells. I'll be describing assays a little later that can actually address these properties. The use of compartments is only there to help us envisage a stem cell system. I'll be delving into this in more detail in a future video on stem cells. But suffices to say that the primary property of stem cells is to proliferate, and all the properties shown on the right, namely self-renewal, primitiveness, potency, and even engraftment, are all dependent on stem cell proliferation. But let's focus on the methodology and how different methodologies can be used to help us understand more about this biological system. Let's first consider the traditional CFU colony forming unit or CFC methodology. This is probably what you're most familiar with. There are 5 main suppliers of methylcellulose media. There's Colony Grow from us, HemoGenix, Methacult from Stem Cell Technologies, Colony Gel from Reach Bio, methylcellulose media from R&D Systems, and HSC CFU from Miltenyi Biotech. All use exactly the same methodology with 35 millimeter Petri dish cultures. All these products contain methylcellulose. They may contain up to 30% FBS, fetal bovine serum. They may be low serum, or they may be serum free. Some will contain growth factor cocktails. Also included will be things like transferrin or reducing agent and medium, etc. Methacult, Colony Gel, the media from R&D Systems, and HSC CFU from Miltenyi all use the same basic protocol that requires dispensing up to 3 milliliters of the reagent into a tube, adding 10% volume of the cell suspension, mixing and dispensing 1 milliliter into each of 2 by 35 millimeter Petri dishes. From a 100 milliliter bottle of reagent, you will get only 30 to 33 samples. However, with Colony Grow, you'll get 44 samples from a 100 milliliter bottle. You'll have more cell populations to measure, as you'll see a little bit later, from up to 8 different species, and they have a comparative or lower price. Here is a cell dose response comparison between the Methacult product H4435 and Colony Grow containing exactly the same cocktail. You should be aware, however, that if you use this Methacult product or a similar product from other companies, you're not actually detecting the CFC GEMM population. By definition, a GEMM stem cell colony must contain megakaryocytes. Since these products do not contain thrombopoietin or TPO, you're not detecting the CFC GEMM population, but actually a more mature stem cell population that does not allow the production of megakaryocytes. So when a company tells you you can detect CFC GEMM, take a moment to consider that this is not actually the case and that it can influence your interpretation of the results. Due to the slightly different formulation of Colony Grow, colonies are usually counted after 10 to 12 days rather than 14, perhaps even 16 days, and produce higher numbers of colonies. Now, many investigators have been told or have read that it is normal to culture the cells with a cocktail of growth factors and differentiate all of the different types of colonies in a single plate. This is shown in the upper graph using Colony Grow that is similar to the Stem Cell Technologies Methacult H4435 product, but the Colony Grow includes TPO to stimulate megakaryopoiesis. 4 human bone marrow donors have been used for this study, so that the variation is apparent. The lower graph shows the number of colonies obtained when each of the individual cell populations are cultured separately. The number of colonies for each cell population are higher than if the cell populations were enumerated in a single plate. In short, by enumerating all of the different types of colonies in a single plate, the actual number of colonies produced from each cell lineage is severely underestimated. Now, this may appear to be a subtle difference, but they can have important consequences, especially in a clinical arena, in drug development, and many other applications. In 1982, I developed a second iteration of the assay. This was a miniaturized assay, which we now called Cameo 4. Cameo 4 is set up in a similar way to the traditional assay, but using just 1/10 of the volume. Instead of doing 2 replicates, it's a quadruplicate culture with each of 0.1 milliliters in separate wells, as you see in the picture here. The methylcellulose is easy to dispense and the colonies are easy to count in 9 to 10 days. There are no expensive dishes to avoid meniscus that are necessary. And the Cameo 4 allows you to do 50 samples per assay kit and includes 50 culture dishes so that you don't have to purchase these separately. And the price is actually similar to Colony Grow. Here you can see there is a direct correlation between Colony Grow and Cameo 4. Many other groups have also compared Cameo 4 to Methocult. Now, before I come to the next iteration of the CFU assay, it's worth considering what some suppliers call standardized reagents. The term standardized reagents used by other companies producing methylcellulose media simply means that the components are manufactured under standardized procedures, usually under GMP or good manufacturing procedures. Standardized reagents does not mean the assay is standardized. A standardized assay is one that includes external standards and controls. The CFU or CFC assay is not a standardized assay. It does not include standards and controls. So intra and inter-laboratory comparisons of results is not possible. However, the CFU or CFC assay can be standardized. And this is where the third iteration of the CFC assay comes into play. I developed Cameo 96 in 2002 for hematotoxicity testing. It was the first methylcellulose clonal hematopoietic cell assay that incorporated 2 endpoints, namely colony counting in a 96-well plate, followed by measuring total colony cell proliferation using an ATP bioluminescence readout. I'll discuss this in more detail in a moment. The assay is prepared in exactly the same way as the traditional CFC assay or Cameo 4 assay. The volumes are similar to those in Cameo 4. You can also use as many replicates as you like. After incubation, the colonies are counted. Total colony proliferation is then measured using the most sensitive and accurate cell proliferation readout available, standardized ATP bioluminescence. Everything you need to perform the assay is included. Since the proliferation step of Cameo 96 is standardized against an external ATP standard and calibrated against external controls, the relative luminescent units, RLU, measured by the instrument can be interpolated into standardized ATP concentrations. When you plot the number of colonies against the ATP concentrations produced, you should get a straight line graph. From the linear regression and correlation between colony counts and the ATP concentration, the number of colony counts can be converted directly into standardized ATP concentrations, thereby standardizing the CFC assay. So this does allow intra and inter-laboratory data comparisons and a comparison of results over time. No other CFC assay has this capability. Now, if you've been using the CFC assay, You have probably been taught that colonies are counted as single colonies, even if there appear to be several colonies together that are delineated by open space, as you see here in the top row of the images. There is, in fact, a considerable amount of movement of the cells during the first 24 hours of culture. When the cells begin to proliferate, they grow as aggregates of cells that gradually get together and form individual colonies that can grow together. Each of these smaller colonies represents units of proliferation that are traditionally considered together as one large colony. The diagrams in the bottom row show how each large colony can be divided into what are called proliferation units. And in this graph, colony cell proliferation was measured on day 7 as relative luminescent units, or RLUs. Colonies were counted on days 10 and 14. The correlations obtained between colony proliferation on day 7 and counts on days 10 and 14 means that proliferation results will predict colony counts. Remember, once aggregates form to produce a colony, the colony will only grow larger due to cell proliferation. And depending on the primitiveness of the cells producing the colony, no new colonies will be formed. But this increase in cell proliferation cannot be measured using a CFC assay. This is what is implied, but is never measured. Therefore, unless you have a method to measure colony growth, the CFC assay is not a proliferation assay, but rather a differentiation assay. So here you have a direct comparison between the CFC products from other suppliers and those from HemoGenix. It gives you a description of all of the parameters that these assays have to offer. And this brings us to the question, why do we need clonal assays anyway? If colony formation relies on cell proliferation, which cannot be measured without a second endpoint, why use a clonal assay? Since differentiation is a default program requiring prior proliferation, and the growth factors used will predict the lineage-specific cells produced, a clonal assay is unnecessary. Why spend time learning and counting colonies when equivalent results can be obtained using a non-clonal instrument-based assay? If results cannot be standardized to compare results over time, why use it? Why spend money on expensive instruments such as image colony counters that can only be used for single purposes when a plate reader is multifunctional? And why spend time and money on proficiency tests for a clonal assay when the results are subjective and show large variabilities between laboratories? These and many other aspects prompted the thought process to consider a world without clonal methylcellulose assays. And that led to what is now called hematopoietic assays via luminescence output, or HALO for short. Now, there are many ways of measuring cell proliferation, and with several of these, it's also possible to measure cell viability. You can measure cell proliferation using radioisotope labeling, but this does not measure cell viability, and it is hazardous. You can count the increase in cell number, and if you're using a viability dye, you can get some information on this parameter also. Measurement of cell proliferation by absorbance using reagents that require reduction by the cell mitochondria are also metabolic viability assays. The same goes for some of the fluorescence proliferation assays, although many of the reagents bind to DNA and do not involve the metabolic process. Finally, we have the bioluminescence assays for ATP and reduction potential. Both of these require mitochondrial function and are therefore also metabolic viability assays. I'm going to focus on the bottom two assays, even though there are the absorbance and fluorescence endpoint assays that I will mention a little bit later. Now, hematopoietic assays via luminescence output are based on the fact that all cells require cellular chemical energy in the form of ATP. When you stimulate or inhibit cell proliferation, the changes in intracellular ATP correlate with these processes. Because ATP is produced in the mitochondria of the cells, it also measures metabolic viability of the cells. The intracellular ATP concentration also correlates with the number of cells being measured. After incubation, a single-step addition of an ATP enumeration reagent lyses the cells and allows the release of the ATP to react with the luciferin luciferase in much the same way as a firefly produces light. Except that here the light is measured in a luminescence plate reader, whose output is relative luminescent units, or RLU. After calibrating the instrument and standardizing the assay, we can then convert or interpolate the RLUs into standardized ATP concentrations. This is the principle that is used to measure colony cell proliferation in the Cameo 96 assay. However, for many applications, the presence of methylcellulose is actually a hindrance. And for this reason, we developed suspension expansion culture technology. Now, suspension expansion culture, or SEC technology, is a methylcellulose-free and also serum-free in vitro culture system for studying lymphohematopoietic cells. SEC technology allowed for the development of the HALO platform with multiple applications: stem cell research, cellular therapy, and toxicology. SEC technology provides the following advantages over methylcellulose assay. First, you can use normal pipettes, so there's no need for syringes and needles. There's greater dispensing accuracy, so you get reduced pipetting error. There's an increased sensitivity, so you can measure as few as 20 to 25 cells. It allows cell-to-cell interaction, and this is very important because it results in a shorter proliferation onset lag time, which is usually within 24 hours, and a shorter culture time, which many of these assays can be completed in 3 to 7 days instead of 10 to 14 days. It's easy to remove the cells for multiplexing with other assay readouts, and it allows for 96 well or 384 well plate high throughput capability and the use of robots for screening applications. And the result is that the methylcellulose clonal assays of Cameo 4 and Cameo 96, as well as Colony Grow, which isn't shown here, all correlate with the results from HALO. There's a simple reason for this. As I've mentioned, cell proliferation always occurs prior to cell differentiation. So if proliferation occurs, we can predict that differentiation will follow, and therefore the two processes must correlate with each other. But there is more to cell proliferation than meets the eye. There are actually two measures or two measurements of cell proliferation. The first is proliferation ability, which is the amount of cell proliferation at a specific dose and point in time. And then there's proliferation potential, which is a predictive measurement of how much proliferation a cell or cell population can undertake. Proliferation potential can only be determined by the slope of the linear regression of a cell dose response curve, using a minimum of 3 points. This diagram shows the cell dose response curves for 7 different stem cell and progenitor cell populations from normal human bone marrow. The vertical line shows the amount of ATP produced from 2,500 cells at the time the cells were cultured. This is the proliferation ability, also called cell quality. You will notice that the dose response lines are in three groups. The upper two lines are for the stem cells, the middle three lines are for progenitor cells, and the bottom three lines are for the lymphopoietic cells and background. Notice how the slope of the dose response lines decrease from stem cells to background. The slope of the line is a measure of proliferation potential of each of the cell populations being measured. The most primitive stem cells exhibit the highest proliferation potential, while the cells with low proliferation ability also demonstrate low proliferation potential. This is an extremely important concept in stem cell biology, and this type of study is very easy to perform with HeLa. This diagram shows all of the different types of stem cell populations we've developed assays for. Even though we have methylcellulose assays for many of these stem cell populations, you will be measuring the differentiation result, rather than directly measuring stem cell proliferation ability and or potential using HeLa. These stem cell populations can now be determined from a number of different species. In addition to HeLa, there are assays with a fluorescence endpoint called HemoFluor and others with an absorbance endpoint called HemoLight, so that you can perform these assays with any plate reader you have in the lab. Notice that the different types of stem cell population are based on the cocktail of growth factors used, just as in the CFC assay. This diagram is a continuation of the previous one, showing all of the progenitor cell populations we have individual assays for, depending on the growth factor cocktail used. Again, in most cases, both clonal and non-clonal assays are available. Here you see how the proliferation ability and proliferation potential decreases from the most primitive lymphohematopoietic stem cells, HPP-SP, to one of the most mature stem cell populations, which we call GEM3. Since proliferation potential is indicative of other stem cell properties and characteristics, it's obvious that you can obtain an enormous amount of information from these assays. In fact, suffices to say that these concepts are used to measure stem cell quality and potency. There are other YouTube videos that discuss stem cell quality and potency in much more detail. Now I'd like to introduce you to a new bioluminescence assay that relies on the reduction potential by mitochondria. It's called HeLa Real-Time. Unlike the ATP assays, HeLa Real-Time uses a reagent that is non-destructive. It can be added right at the very beginning of cell culture or later in the culture, and you can remove the plate at any time and measure the bioluminescence. Here we're measuring the onset and initial exponential proliferation of cryopreserved human bone marrow, what we call SCGEM1 stem cells, over a 5-day period, and comparing the results with the endpoint ATP assay. As you see, proliferation begins within the first 24 hours and steadily increases up to 5 days. This is why the HeLa ATP assay can usually be completed in just 5 days. From the exponential curve fit, we can calculate the doubling time, which is about 1.5 days for each of the batches of human bone marrow cells tested. Since the assay is non-destructive, it is also possible to follow the presence of stem cells and differentiation into different lineages by adding fluorescence-labeled CD markers and performing flow cytometric phenotypic analysis directly on the cells in culture. This table compares HemoGenix assays from hematopoietic cell populations with those from other suppliers. It shows all of the stem, progenitor, and precursor cell populations presently available, together with the growth factor cocktails used to stimulate these populations. You will see that other suppliers only provide a limited number of detectable cell populations for the traditional methylcellulose assays. They do not provide any other assay formats or assays that divide proliferation from the differentiation process, as is the case with HemoGenix. In short, you may not even be aware that HemoGenix has so many different products for hematopoietic stem cell and basic research in cellular therapy. These products provide you with the flexibility you need to tackle virtually any question you might have in cellular hematopoiesis using instruments available in every laboratory. After 50 years of using virtually the same assay, you would expect there to be an update to keep pace with new technology. Only HemoGenix provides you with that new technology. After all, you expect an update for your computer operating system or smartphone to get rid of the bugs so that it works properly and you can do more. It's obvious that other methylcellulose CFC suppliers are not interested in removing the bugs from an assay, improving it, or allowing you to do better science. They know that over the years these bugs have been accepted by everyone else and that you will have to accept them also. But complacency leads neither to innovation nor does it change science and knowledge. The savvy and knowledgeable researcher does not accept this mindset. At HemoGenix, we believe that better assays provide better results, and only non-subjective, instrument-based assays can really provide that sort of quality. Don't be worried that you're not counting colonies. You don't have to, because Cameo 96, Halo, Halo Real-Time, HemoFloor, and HemoLite are going to predict what you would normally see with a traditional methylcellulose assay, but in a more non-subjective and informative manner. So thank you for taking the time to view this video. Connect with us. We always take the time to discuss your options and steer you in the right direction that will make your work a success. Now, this video is accompanied by a more detailed application note on 50 years of colony-forming cell assays and can be downloaded as a PDF file from the HemoGenix website at hemogenix.com. And we'd also appreciate your input. Send us feedback, comments, and questions to info@hemogenix.com. Thank you very much, and bye-bye.